So you all have TVB installed on your computers already. I was told, and I hope you all did that. Okay. So what we're going to do now is we're going to first open the web application. So we're going to go in our TVB folder. For me, I put that in my applications folder. And then for Apple, we have this little app right here. I don't know how it is for Windows or for Linux. But you can just start it by calling your command or your shell script, or I think in Windows it's a bat script, to run the web, to get the web GUI going. There you go. So I'm gonna first go into my applications. Nope. And then I'm just going to call the script, just call TVB start with whatever ending you have on your operating system, and we see it's going to start up the web interface. It's going to use your given default browser. So ideally that would be Chrome. There might pop up some issues with other browsers. I'm using Safari. That worked for me so far. I might have to crop some screen resolutions from time to time to make it look nice. For you, I think the login is the same. Like Julie told us earlier, we can make our own users, but for now we're just using the default user, which is admin as the username and pass as password. Which should lead you, if you haven't played around with it already, which should lead you to the settings page that we've seen on the slide from Julie earlier. Here, just to make sure, the admin username and the password is what we just gave in. The hard disk, the disk space, as we said earlier, if you actually play around or plan on playing around with the GUI interface a lot, you might need to increase that. We don't need this for now, but I don't even know what the default value is that you have in the distribution right now. But you might want to increase that if you're actually planning on running a little bit longer simulations, or just start a lot of different projects with different data that you put in. So you want to increase that. Also, we have the root folder. For me, it's just the TVB in my user folder. This is basically where TVB is going to store all the data that you put in, the simulations that you run, all the project information, etc. is going to go into this folder. Because as we see, we are automatically creating an SQL database there with all the default data that the TVB distribution comes with. And this is it's a pretty nice setup that we have if we're using this GUI web interface. The port is pretty standard, since we are just running stuff local, this doesn't really matter. And again, the same goes for the cluster properties that we have here. The only important thing is that if you have the path variable here for your Octave or MATLAB path, for now just delete it to make sure everything runs smoothly. There can be some issues with the required packages don't really match up on the path variable here. So just delete that and click apply. No, it's still the same password. Doesn't matter. Okay. This should lead you, if it doesn't, to the first tab in our simulator cockpit. So what we see here is the standard GUI interface, and we basically have three main regions. We have the lower region here. We have these different tabs, where we first had the user tab. Where we just put in all the settings that we have. We're now in the project overview. This is basically the entry point to everything. If you're starting with a project, you're starting using the TVB GUI, this is where you start to create your project, or just use the default project as what we are going to do in a second. The next thing, like we've seen before in the lecture about, we have the simulator tab where we will run the simulation. We have the analysis where we have those basic analysis functionality that we've seen before, some of the stuff. that is implemented in TVB that we can just easily run on our stuff via the GUI. No command line is needed for this in here. And then stimulus and connectivity, again, just having those functionalities in their own little tabs. Just because you all, I mean, whoever wants to follow, that is, you all have this. This all worked fine for you. Or is there anybody? Because sometimes we run into issues. On Linux, it might take a little bit longer to load the web interface or something, but everything is running smoothly. Perfect. Okay. That will make this whole thing very quick. The other thing that we have here in our project overview is just the structure of the interface is always the same. If we go through the different tabs, we have the main functionalities in our tab that we have present here. Just the operations that we've seen before is all the operations that we perform and using this interface are stored here. Even if we perform operations in the command line and later on import this project into our GUI, we will find them here. So images, everything, that's pretty self-explanatory. So what we want to do now is we want to go and check out the projects that we have and that we want to do some simulations with or create our brain models. So what we do is we click on the list of projects, and for you, I think this is only the default project. I played around with it a little bit, so we have the Node 2. This is what we're going to do later. We're going to do again, actually. For this, an important thing is this is a nice overview that tells us the status of our project. We see here in the top left corner is the 1 because the project ID 1 is the one I'm having currently selected. So if I switch into the other tabs and simulator analysis, is still I'm going to be in that project. We just select it by starring. It's pretty easy. And if you click on it, we get a short overview of what we already did with that project. And especially if we have multiple things running at the same time, this can give us an overview, especially if something crashed. Everything, well, most of the things are really nicely color-coded in this web GUI. So is the overview. So we see here in the default project, I already ran 114 operations that all went perfectly well. Nothing is running right now, which would be the blue. Obviously, nothing failed ever with me running that project. So we have 0 failed operations. Note to this, if we go into the operations overview, we can delete operations that failed, for example. So we have the nice little, the nice little 0 here. And we see an overview of the size of all the data that we have put into that. So what we want to do now is we want to, with this hands-on session, we want to get to the point to start our own simulation. So since this is an individualized brain simulation, we need to have actual data that we base this in the simulation on. So the first thing we're going to do is we're going to look at all the data that we have available in our default project. This data is already loaded in. This, even for you, the default project should already show some operations, and those were just loading in this data, which comes with the basic, with the distribution that you downloaded, which is pretty nice because this way you can just start right from the get-go and get some simulations going. Here we see the overview of what we have present. I think you should probably have a little bit less than I do, but just gonna go really quickly what it is that we have here. Again, we have a nice color coding. This is, I don't know if this is visible on the screen. Yes. But you should see this on you as well. We have the different data types which are color-coded in the TVB GUI. Green is the raw data that we import or have default-wise loaded into our TVB. This is something, for instance, like the cortical surface, which is just the raw quotation marks data that we put in here. This is data that we get from pre-processing that we have done before putting this into our simulation software. We think we're going to have a session or lecture on the pre-processing to get to this point tomorrow on Wednesday, because it's nice to have this all loaded in here, but getting to that point to actually have this pretty surface extracted, etc., is a whole different story. So when we click on it, we see already a nice little overview, which is not as important since this is default data that we have, and this is obviously clean and very nicely very nicely prepared. But if you start doing something like importing your own connectivity, importing your own data, this can already be a first instance of something of a sanity check to just look, is this something that we expect? Like the number of edges that we see, does the processing work in a way that we can use it for TVB? And what we see here is just the metadata overview. And one important or interesting thing, for instance, is the surface type, which called us, okay, this is a critical surface. This is a surface data type. TVB has data types for every possible type that we would need in this simulation framework, and specific properties, but we'll get to that a little bit later. One very important thing in TVB is, if you start getting into it a little bit more, is the so-called unique global identifier that we can see here. So this surface file that we were looking at has a unique identifier for everything that we do within TVB. This doesn't matter if we're using this project within the web GUI or we're using this project running command line interfaces. We have this global identifier. This is a very nice property, because if we later on start doing another project with the subject or whatever, and we want to upload our data, we will get an error message telling us that this identifier is already present. So what we want to do with this is we don't keep uploading the same data into our database and just trashing it with too much data. We're just going to link data that we already have in our project to the projects that we have. So we just go into link and say, okay, just link this data into our new project. If we ever come to this case. We don't do this now because the other one is mouse and this is a human surface. This doesn't really make sense for us. The other very nice thing that this web GUI has, and why, even if you're using the command line interfaces later on, if you're getting deeper into it, is the visualization capabilities. And this is for the surface visualizer, for the surface very simple. It's just take a look at the actual surface, and we get some of these nice pictures that we have seen before on the slides when looking at the TVB. So this is a pretty straightforward just visualization of the surface that we had. I just clicked that a little bit too fast, but this is just the back button to get back to our overview. So this is the raw data that we have, which is color-coded in green. The next type of data that we have, or data type that we have color-coded, is yellow, which is called the adjacent data type, which basically means that this data exists in connection or based on data that we already have present in this project. For example, looking at the surface EEG projections for our given surface. So if we look at this yellow data type, we have this projection surface to the surface that we just looked at. And we don't need to look at all these different things, I guess. The other main input that we had beside the surface is our connectivity. We can take a look at this really quick, because this are some of those nice pictures again that we've seen before. This is the data that we extracted from our diffusion weighted imaging that we get to the connectome, and we can actually look at it. What is the basis? So much to it is messing up the way it looks. Yeah, there we go. I don't know if that happens to you. For me, I just zoom out and it works again. And this is the connectivity matrix that we're gonna base our simulation on later on. These are, in our case, we have the two informations available. We, for one, have the weights, the weight matrix representing our connectivity, and we have the tract. The one representing basically the number of tracts connecting these regions, and the other one being the distance of these regions, which can be important when considering delay times in modeling the different interactions of regions in our model later on. And when I said we have nice visualization capabilities, this means we have this 3D connectivity representation, and we can also just look at all the connectivities that we have present, all the connections. That's just by showing and hiding them. The same way it goes if we just if we go on and we don't look at healthy brains, for example, but look at pathological brains, we can say, yeah, we want to focus on specific nodes in our network. Then we can select the thing in networks. We see it already popping out as in our matrix. If we go over it, mouse over as the initiating and the receiving node in this network, and then we can, for example, just also toggle all the incoming connections that we see to this node, and then we see how it is connected. Did that not work for you? This does work for everybody. If you just try it out a little bit.
How did you get the pop-up?
The pop-up is you just select the region or connection in the connector.
You don't have a rate
Thank you.
check page.
Yeah, I don't know. Maybe it's down here. Oh, there. It's because my screen's too small.
Are you following along?
Yeah.
Wait, and the weight matrix doesn't show for you?
It does. My screen's small, so it is beneath.
Okay, because I had that, and then it just zoomed out, and it just popped out on the right side of it.
Uh,
Like I just pressed Command minus to zoom out in the browser,
Yeah.
and then
Yeah,
it just
that works.
perfect. Yeah, this is, I don't know, this is Safari. Like I said, you sometimes have a little bit of an issue with some of these things.
Can you just, what did you do?
To get there?
No, I don't remember. What did you, no, not to get.
So-
Just what you were doing.
Just the selection itself was just to have a look at the
Yeah.
connectivity that we have present. But for everybody else, it worked. Good follow. Okay. Okay, I don't know why this is there. There we go. What is also interesting is here that, like I said before, if we want to look at pathological brains, for instance, imagine we know that certain areas are infected because of a stroke or whatever, and we have a lesion in the brain. We can even select this region and say we want to cut out all the ingoing or outgoing connections that we have to the specific area.
Is that not? Sorry, can I?
Yeah.
ask again?
Sure, yeah, sure. So, go ahead.
Isn't that solved by computing the matrix, the connectivity matrix? I mean, you ask the lesion mask to the parcellation, and then you can exclude it, right?
That is one approach. Yes, I mean, for instance, for this, you would have to have a lesion mask present. So you would have to have a neurologist first go in, probably paint the lesion mask in whatever, in the T2 or in the FLAIR image or whatever, and then you can apply that when computing the connectome based on the tractography. But this is actually a research project that we're doing right now. The question is how to deal with stroke lesions in this context, because a lot of the time, if we have the connectome built on the fusion rate imaging, we still have viable tracks running through the lesion. So the question is, do you cut it out by just giving the lesion, by masking the image and lesion mask and don't even computing it in the matrix, or compute the matrix like we have it right now and just afterwards apply specific filtering to that area to say, okay, we want to cut out the whole lesion, etc. So this is
You're using FreeSurfer for that.
yes, you use it. It's FreeSurfer based. FreeSurfer, FSL, a lot of stuff.
A career from your side,
Hmm?
which is better.
This is, I could not answer this because this is still ongoing. A lot of research these days goes into the direction to just completely delete it and basically just replace the lesion tissue with zeros in the matrix. But there is argument, especially if you have bigger lesions, that you still have viable tracks going through. So maybe just the weighting of that or something like this could be more appropriate. But this is ongoing research. It's not conclusive for that part yet. Okay, so this is just to have a look at the connectivity that we're gonna base our little simulation on later on. The next data type is not present in this default right now. It would be color-coded in blue and represents results that we have of an analyzer that we run within the TVB framework. So like I said, we have these nice analysis functionalities like independent component analysis, functional connectivity dynamics, which you're going to look at again a little bit later. And stuff like this, we have that implemented. So if we run the analysis, we're going to get another data type, which is color-coded in blue. The next one would be red. That is a very important one because this is the time series data type. So this is where we actually have our simulation results. We also see this one should be purple. It's probably an account, but this is just the data type if you save data or applications that you made to your data. So for instance, for me, this is local connectivity because I went into the given connectome and I added new connections and saved it here via local connectivity. This could be a stimulus that you computed that you want to apply later on again. This could be all these kinds of things that you actually perform inside of the TVB. This has its own data type again. So why they have all these data types, we're going to get to this a little bit in more depth with the mouse simulation that we do later on. But this is basically the straightforward how much we have so far. Do we have anything else that we want to look at in terms of? We also have in our default project, what we're going to simulate later on is EEG data or EEG time series. So what we have here also in our raw data is the actual positioning of the sensors of our EEG cap. And again, if you just look at this, just to have a visualization of those sensors, we again zoom out a little bit, and we can actually see the positioning of our sensors. To this, we again have a yellow data type, which basically is the mapping of these sensors to the surface that we had looked earlier. So it's this adjacent data that we see combining the two raw data types that we have, so that at the end we can simulate EEG data on our surface and compare it with actual EEG data if we have that connected from these positions that we have and these localizations of the sensors in our brain. I think this is everything we need if we want to get a simulation started. And for this, we would go into the simulator network, and I think Paul is going to go and look at the parameters that we need, how to get our first simulation going.